Thank you. First of all, I think thanks to the organizer and the chair create this opportunity for me stay here, report some work from our group. So you cannot find any information from your booklet. I'm a guest speaker. So, okay, so yeah, you cannot find because I think it's maybe later on they will add. But hopefully this presentation not cost you, waste your time. So today I will talk about some stem cells and bioengineering with diabetes. So as the title you can see, we use the bioengineering approach with the stem cells to try to create some artificial human islets for immunorejection free and human diabetes treatment. So as everybody knows, the diabetes become like a cold. The WHO reported in 2050, every 3 people will 1 get diabetes. It's a very become very popular disease, but cure is still very limited in the market. Beside of the insulin, nothing very effective. So islet transplantation actually offered a very opportunity, the way to therapy for diabetes treatment. But the folks faced very severe stubborn barrier. So I don't. Do you find any hospitals do the regular islet transplantation? No. This process, transplantation, is very easy. However, the islet itself, human islet, they are very difficult survival and working. So the problem is the islet cannot survive longer and stop the regeneration. So currently, the mountain protocols is used mostly in the world, but it's still not very so efficiently. This is basically is the diagram introduce what is the islet transplantation is. The first will get the donate islet or donate the pancreas isolated islet, then transplant into this liver tissues. They try to let them cure diabetes, but the success for rate is very low until to the recently updated. The 2014 is still not so high survival rate for long term, almost no long term treatment. So bigger surgery process, but no long term treatment. The problem is islets function and survival. Islets, they are given to transfusion into the in vivos, but they died soon. So in our group, we are already working in this almost 10 years. So we proposed there are some way can be support human islet survival and protect them from loss of function. So what do we do? We use the human islet with the human bone marrow, allogenic bone marrow. So here is the islet with the bone marrow. Here's the islet only. After 6-day culture, do you see any difference? This islet with the bone marrow, they looks like more structure, keep this islet micro organ structure. But this one, see, the cells all open the windows, very exciting, come out. Finally become the monolayer, become the monolayer cells. Okay, here. But after same times culture. Oh, thank you. Yeah, thank you. Yeah, after same time culture, see, this islet become bigger, the 3D structure. So what is the function? So we measure the function. Say, here is the islets only. They are, from beginning, you didn't see any significant difference. After 1 month, say, islets only gradually lost the function, okay, in more than 6 months. But islets with the bone marrow, the end of this time, they will more regeneration, rebound their function. Also, we use high glucose challenge. Give the glucose. Say this islet responds to this, releasing more insulin. Actually, the similar model as the previously, the releasing of the data. So we proposed bone marrow through this multiple mechanism repair the injury the cells through this paracrine, include the many microRNA, other factors, and even differentiation itself to support the human islet. So this is another tissue we generated after 6 months with the human islet with the bone marrow. See, they generate bigger tissues, more than 1 centimeter long. There are some vessels in there. So we do the HE staining. They keep this islet structure and use the insulin immunohistochemistry to try to identify the insulin cells. The red is the insulin cells. The green is a glucagon. So blue is a nucleus. So we see a lot of insulin cells in the human islet. So also function is very significant difference. Say bone marrow have the significantly high insulin releasing, and islets almost lost the function. However, at the high glucose in the 30 minutes or 60 minutes, also showing significant difference. That means these human islets, if they are in vivo, we suppose they should be working very well to support your body, reduce your blood glucose. Okay, further works we find is here is the islet only. This islet with the bone marrow. We can see this structure are different, right? Say we'll put these parts in the sections to analyze what is the structure is. Is green is the green is the CD31 endothelial cells. Red is the insulin cells. So we say they are the beta cells surrounding with the endothelial cells. The vascularization very well. Okay, so also we do this protein array evaluation. Say what is this co-culture have factors. We find all this group vascularization relative the factors like VEGF, PDGF, the KGF, and angio. This all significantly higher, but they are up and down is for long term, like more than 6 months. So we use the deprivation method. Try to this is the islet only couch is day 2, and day 56 is also, as with previous data, it's consistent. So it's not good. But with the bone marrow, they were very good. If we deplete the KGF, less than this whole bone marrow treatment. Okay, if we deplete the PDGF and the KGF and the PDGF, the human islet looks not so healthy, especially when we deplete the VEGF. That results very significant difference. So it means that VEGF maybe plays a significant role in the for the human bone marrow support the islets. So this is the quantity result. So what is the population in the human bone marrow to support the human islet? That's the bigger question. Everyone, when we're starting with this project, they asked, what cell population plays significant roles? We did some work. We get mesenchymal cells, endothelial cells, and bone marrow whole cells. We find these three populations for support the human islet function, no significant difference. This is insulin releasing. However, we find a difference, okay? For the beta cell regeneration, beta cell quantity, the beta cell numbers, we find is endothelium and the mesenchymal cells, yes, a little significant, a little improved for the islet beta cell regeneration, but it's a bone marrow significant difference. So I think a lot of papers already outside, say, as mesenchymal cells, endothelium cell, progenitor, there can be differentiation to the many cells. But for islet endocrine cells, it's different. So we try to find, first of all, islet beta cell itself, they can regenerate by themselves. So in order to answer the questions, we use this Krelok's the approach. Krelok's mostly, as I just reviewed with you, maybe you can understand. There's two vectors. One is Kre, the enzyme. One is LOXP. They insert of the red color and the green color. So the Kre is the enzyme. They connect with the insulin promoter. As soon as the cells become the insulin, they will activate the Kre. The Kre is like a young gentleman. When the activities they see goes red color there, they will special come here. They will cut this part. In other words, if the cells have the insulin, they will activate the Kre. They will cut this red. The cells will become green. So what is happening with a real experiment when we transfer this Krelok system in the cells, islets, human islets, we find there are some few islets positive beta cells. Some is non-insulin positive cells. Okay, after the cultures, in the islets only, we see gradually reduced the beta cells, insulin cells. However, with the bone marrow, we see the more cells grow up, more green color become. So as we do this immunofluorescence to confirm this green cells is insulin cells. So then we calculate the quantity of all the cells. So we find a very interesting thing is actually human islets beta cells itself can be regeneration. However, need a special condition. Say first, less than 1 week, they are all grew up. Okay, islets only, islet with bone marrow. But after almost 1 week, the regeneration, the capability in the islet lost. This is a human islet. Okay, so then bone marrow able to sustain this human islet beta cells self-regeneration. That is a big find. We find that this will be useful for human diabetes therapy. So then, how we can know this? The in vitro system can be working in vivo. So we use this pre-cultured the human islet transplanted into the kidney capsule. Okay. Yeah, this is a kidney capsule, injections kidney capsules. Okay, so why the kidneys? Because the kidney capsule is very limited tissues. The cells, the islets, as soon as they transfer into there, the barrier to move other place. Then after that, we can take this a kidney. We say what is a human islet function is. Say, here is the result. Say, this is an islet only. They cannot survive too long. They will have to sacrifice because of high glucose diabetes. However, with the bone marrow cultured islet, they support this blood glucose in the normal level. When the day is 4 months, we took this kidney off, which is take the human islet off. We see glucose significantly increased. So no human islet working on there. Another evidence supported this human islet works is we find that in vivo the blood test have the human insulin. So this experiment very supportive for our hypothesis. The human bone marrow, allogenic bone marrow can support human islet as a therapeutic way we can use that. So we did a little the question seeking. So why this bone marrow cultured islet will be survival better? You see, here is the transplant side is a capsule, right? We can see islet only in capsule is no positive. This is the positive insulin staining, okay? No positive. The cells all go into, they are cells, but they are not insulin function cells. However, in bone marrow with islet, they are the function cell. Most interesting things, these cells in vivo after so many months, they are traveling. Hey, they love to from Rome to the Milan. So here's the measuring. So this is the kidney, the measuring area. That's with a lot of vessels and as a support area. See, the islet cells migration, the medium, they are support more, for releasing more the insulin easily. Oh. Okay, so 5 minutes. Okay, so then the in vivo we approved this, supported this idea. Then immunorejection still the questions needed to overcome, right? Say we developed the bioengineering encapsulation project. Say the islet inside, the older bone marrow in the outside, other cells cannot get in. So then we try to find what is the optimize the bone marrow and the islet inside. We find is 5 million bone marrow with islet at the best function with the encapsulation. Then we test in vivo. So we find is a very positive result. And this is diabetes control, normal control, the islet encapsulation only, this islet with bone marrow. We say very good result. So what happens? See, here's the islet only. Here, the islet with the bone marrow. You see what it is? Generate a lot of the vessels in vivo. Okay, they support of this islet. Okay, use immunofluorescence analyze. We find a lot of positive insulin cells with vascular tissues. Okay, so this is another result. After 6 months, we took this tissue off, islet and islet with bone marrow. We find is a significant difference, still functional. So this is big vessels in the islet clustered tissues. So support the islet. Okay, so future work needed to develop clinical studies. Okay, clinical trial. So summarize. Bone marrow increased human islet survival and function. In vitro, not only, also in vivo. They are basically is support human islet with more angiogenesis and regeneration. So the encapsulation, the microenvironment creates is a suitable growth condition for islet survival in vivo and in vitro. So this work can be applied for clinical. We have the patent, US patent, Europe patent, and Canada or different countries' patents. If anyone interested, so we can working together make this happen in humans. So this is a group of my working with this project and a lot of people. So this is the funding conditions, funding resource we supported from NIH and the Juvenile Diabetes Research Foundation, and also our hospital. Okay, thank you. I'd like to take questions.
Thank you. Is there any question?
Yeah, go ahead.
So what is the special property you think that comes with the bone marrow cells that doesn't come with other stem cells that is allowing these beta cells to survive?
Yeah, actually, I oh, okay. So is there any other population from bone marrow sustaining human islets?
No, what is the special property that bone marrow cells that allows?
What is the special population sustaining?
Property of bone marrow cells.
Property is the population, right? No?
What's the special about the bone marrow cells?
Oh, okay. So what is the special bone marrow cells support or influence the human islet beta cell function, right? See, as our results are showing, we test actually at more than 5 different populations. None of any single one as working as good as a whole bone marrow. Why? Because the human islets, they are going to lose function. There are 3 stages. Number 1 is early stage. They lost function just by apoptosis. They just by apoptosis. That's a lot of people already find after transplantation. Number 2, they lost the capability for regeneration, self-regeneration. If you cannot self-regeneration, no cells can be longevity. They have to have some regeneration, replace some lost cells. Okay, so what the cells will support bone marrow population property, support islet longevity, include the regeneration and function. So we proposed the early stage, the bone marrow through this paracrine, they stopped apoptosis, protect the cells loss. Okay, then later on, after 1 month, the bone marrow stem cells and the paracrine and the differentiation will support the human islet beta cells regeneration. So that's why we see after 6 months, we see the cells starting to function to rebound. Also see the tissue is regenerate very rapidly. That is a different stage. Maybe we can use different cell population. But for the project need like a whole